Eu começo, eu falo
Tá.
primeiro, tá?
Ok.
Boa noite a todos. Para quem não me conhece ainda, meu nome é Bárbara, eu sou farmacêutica, sou doutora em Ciências Farmacêuticas e sou coordenadora dos cursos de Farmácia e de Estética e Cosmética da FASF. Gostaria de agradecer a presença de todos, nossos convidados, todos os participantes do primeiro Seminário Internacional de Ciências Farmacêuticas da FASF, nesse momento em que fomos surpreendidos por um vírus, uma doença, uma pandemia, e a ciência, mais do que nunca, ganhou notoriedade. Cada ser humano está apostando nesse momento todas as suas expectativas na ciência, para que essa situação seja logo resolvida e para que possamos ter a nossa tão sonhada vida de volta. Cada pesquisador, guardadas as devidas proporções, passou a se dedicar mais e mais ao seu trabalho, na busca das respostas corretas e das melhores soluções para esse problema que estamos vivendo. A FASF, em seu compromisso com a qualidade de ensino, pautada na tríade ensino, pesquisa e extensão, apoia a ciência em todos os seus níveis. E esse evento é mais uma das etapas no longo caminho percorrido na maneira FASF de educar, focado em formar profissionais não só bem-sucedidos nas suas profissões, mas também humanistas, críticos e reflexivos, e capazes em suas áreas de atuação. É nesse contexto que temos hoje a grande honra de receber notórios pesquisadores. Dentre os inúmeros agradecimentos que temos a fazer essa noite, eu destaco o professor Thales Renato, que foi o idealizador e organizador desse evento, Dr. Marcelo, que é um solícito, né, foi o responsável por nos conectar com a Universidade Laval e viabilizou, assim, o evento. A Associação Brasileira Beneficente de Apoio ao Alérgico e ao Canada Gene Therapy Group, pelo apoio ao evento. A Wizard Luz, Gabriela e Matheus, por facilitar o nosso entendimento do assunto. E, é claro, o professor Jacques, Gabriel, professor Bandeira, por nos brindarem com suas apresentações essa noite. Sem mais delongas, ansiosa pelo início dos trabalhos dessa
Boa noite a todos. Eu vou me apresentar a vocês. Meu nome é Luiz Werber-Bandeira. Eu sou médico. Eu sou PhD pela Universidade Federal de Medicina, em Medicina. E tenho pós-doutorado em Imunogenética pela Fiocruz Rio de Janeiro. Eu chefio os serviços de Imunologia e Alergia Clínica Experimental da Santa Casa do Rio de Janeiro, que provavelmente esse serviço é o serviço mais antigo do Brasil. Foi criado pelo professor Clementino Fraga Filho, e seu primeiro chefe, quem eu fui assistente, foi o professor Antônio de Oliveira Lima. A nossa palestra, eu resolvi fazê-la em português e tem o objetivo de transmitir para vocês os conceitos básicos e primordial do Crispr-Cas9. Eu vou falando em português, mas eu vou fazer uma saudação ao professor Jacques Tremblay em inglês e ao Gabriel. Depois eu volto para o português. Eu quero agradecer muito a organização desse evento, ao professor Thales Renato Carvalho e ao Dr. Marcelo Bossuat. Good evening, Professor Jacques Tremblay. It's a great honor to hear your words about CRISPR. CRISPR, I could say CRISPR is an intriguing chapter of medicine. It's an intriguing chapter of life. And I'm sure that this meeting will be success. And I'd like to thank you and Dr. Gabriel Lamothe for accepting the invitation. Now I'm going to share my screen to begin my talk. Voltando ao inglês, nós temos hoje a grande honra de moderar um Seminário Internacional de Ciências Farmacêuticas, a perspectiva diagnóstica e terapêutica utilizando a tecnologia Crispr. Os organizadores desse evento é a Faculdade de Filosofia e Ciências e Letras do Alto São Francisco, o professor Thales Renato Ferreira Carvalho e Canadá Gene Therapy. Nós temos os palestrantes, o professor Jacques, professor do Departamento de Medicina Molecular de Laval, Quebec, Canadá. Ele falará sobre o uso do Crispr para tratar doenças hereditárias. O segundo palestrante será o Dr. Gabriel Lamothe, que é do Laboratório de Medicina Molecular da Université Laval, em Quebec, Canadá, que falará sobre o uso da tecnologia derivada do CRISPR para detectar infecções virais. E eu como moderador. O apoio do Departamento de Medicina Molecular da Universidade Laval, Quebec, Canadá, a Faculdade de Filosofia, Ciências e Letras do Alto São Francisco, a Associação Brasileira Beneficente de Apoio ao Alérgico, a Canadá Gene Therapy, a Associação de Saúde e Sustentabilidade, ASAS, que é uma associação nossa com objetivos sociais na medicina, no Serviço de Imunologia Clínica e Alergia da Santa Casa do Rio de Janeiro e a Clínica Imunologia. Então, eu vou começar a falar com vocês, são assuntos tentando explicar de maneira bem pragmática e dos conceitos fundamentais do CRISPR-Cas9. Então, o sistema CRISPR, que vem do inglês Cluster Irregular Interspace Short Palindromic Repeats, são repetições palindrômicas. Eu vou falar para vocês também o que significa essa palavra palindrômica. Ou seja, repetições palindrômicas curtas, agrupadas e regularmente interespaçadas. Consiste em pequenas porções de DNA bacteriano compostas por repetições de nucleotídeos. Então, no DNA bacteriano, e no nosso também, depois foi descoberto, nós temos essas repetições curtas. E essa descoberta do CRISPR, ela começou a ser falada ainda em 1986. Em 2010, começou a aparecer as pesquisas sobre o CRISPR e o nosso serviço de Imunologia da Santa Casa, com um acordo de cooperação científica, nós começamos a trabalhar com isso há 5 anos e hoje nós temos, junto com o professor Jacques Tremblay e a Fiocruz, do Rio de Janeiro, nós estamos fazendo uma repetição de uma técnica que utiliza o CRISPR para detecção muito mais rápida, muito mais barata do vírus, inclusive do coronavírus beta, da Covid-19. E estamos aguardando também a vinda bem breve do Gabriel Lamothe, que é um dos palestrantes, para participar dessa repetição dessa técnica do CRISPR feita pelo professor Jacques Tremblay. Então, o sistema CRISPR, ele significa, no nosso DNA, são repetições curtas e agrupadas regularmente na porção do DNA bacteriano composto de repetições nucleotídeas. Vamos à frente que vocês vão entender mais. Cada uma dessas repetições encontra-se adjacente a um protoespaçador, um espaçador de DNA, que corresponde a uma região não codificada inserida no DNA bacteriano após o contato com genomas invasores provenientes de vírus, bacteriófagos e plasmídeos. Ou seja, a detecção desse conjunto CRISPR-Cas9 foi feita numa bactéria que se defende da invasão dos bacteriófagos. E é muito interessante, eu gosto de dizer que só Deus mesmo explicaria isso. A bactéria, então, no seu DNA, ele tem esse conjunto de repetições de nucleotídeos, e ele é capaz de induzir a formação de uma enzima Cas9 que vai lisar o DNA, o RNA do vírus e incorpora esse pedaço de DNA, essa faixa de DNA ou do RNA no seu próprio DNA. Então, eles serão os protoespaçadores e eles ficam localizados adjacente a esse CRISPR da bactéria. Na próxima vez que essa bactéria entrar em contato com o vírus, então o DNA dela é capaz de detectar esse protoespaçador que está no DNA, que é a sequência de base nitrogenada do vírus. E com isso ela sabe que está sendo agredida, já tem uma memória, e ela então, o DNA dela, ela induz a formação de um RNA que vai produzir uma enzima para lisar o vírus, e essa enzima é o Cas9. É muito interessante que esse protoespaçador, que é um pedaço do RNA, o DNA do vírus, ele não tem penetrância, por isso que é uma região não codificante. Do lado dele que fica a sequência do CRISPR. A transcrição do locus do CRISPR resulta em pequenos fragmentos de RNA, com capacidade de desenhar o reconhecimento de um DNA exógeno específico e atuar como um guia de modo a orientar a nuclease caspase, que irá promover a clivagem e, consequentemente, eliminar do DNA invasor, caso esse entre novamente em contato com a bactéria, atuando como importante mecanismo de defesa contra DNAs invasores. Então, é justamente isso. Quando o CRISPR, que tem essa repetição de bases nitrogenadas, que está do lado do RNA, o DNA, que foi trazido lisado pela caspase 9 e incorporado no DNA da bactéria. Quando o vírus reentra nessa bactéria, essa bactéria, então, através do CRISPR e do fragmento do RNA ou DNA do vírus, ela detecta o vírus, produz RNA, e esse RNA produz a caspase 9, que vai destruir esse vírus. O CRISPR, então, ele é repetições palindrômicas, vou explicar para vocês o que é isso, de pequenas repetições palindrômicas que são interespassadas regularmente. Tem um desenho que eu vou explicar. O CRISPR é uma família de sequências de DNA, bases nitrogenadas, encontrada no organismo procariontes, no DNA, em bactérias e na arqueia. Algum tempo atrás, pensaram que a arqueia fosse uma bactéria, mas sabe que não é uma bactéria, e é muito interessante que é a estrutura mais antiga encontrada na Terra. E essas sequências são derivados do DNA do vírus. Então, essa sequência de bases nitrogenadas que está do lado do CRISPR, ela é derivada do bacteriófago que infectou essa bactéria. Então, o CRISPR é capaz de detectar uma nova invasão e capaz de produzir o Cas9. Na realidade, então, o CRISPR, o sistema CRISPR, é um sistema imune da bactéria, do procariótico, que confere resistência a elementos genéticos estranhos e assim se apresenta dentro do plasmídio ou dos fagos. que fornece, então, uma forma de imunidade adquirida a essa bactéria. Então, o que é uma sequência palindrômica? Essa sequência palindrômica, ela só não existe somente nas bactérias, não. Esse nome palindrômico também serve para determinadas palavras. O que é um palindrômico? É uma palavra ou uma sequência de aminoácidos, bases nitrogenadas, que você lendo do início para o fim, ou do fim para o início, é a mesma coisa. Então, isso aí, quando você tem o nome de uma pessoa, Ana, esse nome é palindrômico. Tanto faz você ler do sufixo, do prefixo, e você tem o mesmo nome. E é justamente isso que acontece nas sequências palindrômicas do DNA. Vocês podem ver aqui que você tem da parte do 5 para o 3' ou do 5 para o 3', eu tenho uma sequência pequena de bases nitrogenadas que elas estão GAATTC e a outra GAATTC. É a mesma coisa, tanto faz. Então, por isso, tanto faz você ler esse Crispr, esse conjunto de pequenas bases nitrogenadas, da esquerda para a direita ou da direita para a esquerda. Essa sequência, que existe também no ser humano, ela fica no DNA, e do lado dela que fica aquela sequência na bactéria, que fica do lado do CRISPR dela, no DNA dela, aquela sequência de bases nitrogenadas do vírus. E com isso, quando esse vírus... E essa sequência, é muito importante que essa sequência de base nitrogenada do vírus, ela não tem penetrância genética, ela não induz proteína, não induz enzima, ela não tem atividade genética. Então, o CRISPR, que junto com essa sequência do vírus, sem atividade genética, que vai detectar a nova invasão viral e será capaz, então, de produzir enzimas que vão destruir o vírus. A sequência palindrômica, aqui você vê ACCA, e aqui você tem ACAA. Aqui também TTGT e na outra faixa, TTGT. Então, tanto faz eu ler daqui para cá ou daqui para lá. Então, isso que é o que significa o nome palindrômico. Então, terminando, porque o meu objetivo, não sei se eu estou sendo claro, é dar para vocês os fundamentos básicos, essenciais do significado de CRISPR, dessa ação bacteriana sobre o vírus, para que vocês possam entender tanto a palestra do professor Jacques. E o professor Jacques, eu quero deixar claro que ele foi um dos primeiros pesquisadores do mundo a trabalhar com CRISPR. E lá que eu encaminhei, mandei para a Universidade de Laval, para o laboratório dele, o nosso coordenador científico, o Dr. Alberto Soares, da Fiocruz. Ele ficou um período lá no laboratório do professor Tremblay, aprendendo essa técnica de utilização do CRISPR como terapia. Então, você vê aqui, você tem a membrana celular da bactéria, você tem aqui o bacteriófago, ele entra, tem o DNA viral com cadeia dupla, e aqui está o DNA da bactéria, aqui nós temos a série CRISPR da bactéria no DNA dela, ela vai produzir caspases e vai lisar especificamente aquilo que dá a ela a capacidade de colocar do lado da série CRISPR, esse conjunto de bases nitrogenadas do DNA viral. Por isso que eu disse para vocês que essa ação aqui é uma ação milagrosa, né? Como é que se explica que ela consegue, através das caspases, quebrar o DNA do vírus, especificamente trazer para incorporar ao DNA dela. Essa sequência aqui do vírus, ela é que faz esse espaço, né? Ela não tem penetrância genética. Quando esse vírus entrar novamente, o que vai acontecer? O DNA dele sensibiliza a bactéria, ela tem aqui esse CRISPR, que aqui tem aquela sequência que se repete tanto da esquerda ou para a direita, que eu posso ler, mas aqui que é importante, ela incorporou no seu DNA a sequência de base nitrogenada do vírus. Então, isso dá a ela agora uma memória adquirida, porque ela não é uma memória inata, ela foi adquirida a bactéria entrando em contato com o vírus. Então, com isso, ela vai produzir as caspases, primeiro vai produzir o RNA, que vai produzir o complexo de caspase 9, que vai direcionar o vírus e destruir o vírus. Então, isso é o que eu tentei, espero que eu tenha sido claro na explicação desse mecanismo do Crispr e dos conceitos fundamentais. Depois nós vamos abrir a pergunta, vocês podem perguntar. E agora eu vou voltar, peço permissão para vocês, para voltar a falar inglês, só para começar ou iniciar a chamar os professores. Well, Professor Jacques Tremblay, you can now start your speech. Welcome to the symposium.
Thank you for the invitation. Let me start my presentation. Research work done in my laboratory using the CRISPR-Cas9 technology. Today's presentation will be divided in two parts. I will first present the use of the CRISPR-Cas9 technology to treat hereditary disease, and my graduate student, Gabriel Lamothe, will present how the CRISPR-Cas9 derived technology may be used to detect viral infection. The word CRISPR stands for Clustered Regulatory Interspace Short Palindromic Repeats. CRISPR-Cas9 technology is derived from research on bacteria. It was initially discovered in 2005 that the CRISPR sequence contained viral sequence, and yet there was an hypothesis that CRISPR-Cas9 is the bacterial immune system. It's only in 2013 that it was discovered that the CRISPR-Cas9 may be used to induce specific modification of the human genome, and this started a big explosion of articles. Indeed, the CRISPR-Cas9 system is used by bacteria to kill bacteriophage. Bacteriophage are extremely abundant. They're 10 times more abundant than bacteria, and there is a war that is ongoing between bacteria and bacteriophage since millions of years. It was initially discovered that there are in the genome of bacteria constant repeated sequence. Researchers were wondering what these repeated sequences were for, and then they discovered that the sequence between the constant repeat were sequences that were from the genome of various bacteriophage. In fact, what normally happens is when a bacteriophage infects the bacteria, normally the bacteriophage wins, the bacteria is dead. But sometimes you have a defective virus that will infect without killing the bacteria. Bacteria will then be able to acquire some part of the sequence of the virus, and it will store that into its genome, and it will be ready when it meets such bacteriophage again in the future. From the DNA in the CRISPR array, bacteria will first express a pre-CR RNA, and then there will be a complex that will be made between the tracker RNA, CR RNA, and the Cas9 protein. This tracker RNA, CR RNA, Cas9 protein complex will then bind with the bacteriophage DNA and induce a cut of the bacteriophage DNA to essentially kill the virus. To bind to the DNA, the Cas9 protein requires the presence of a protospacer adjacent motif, which is simply NJJ for the Streptococcus pyogenes tab. Then there is the CRRNA, which contains a variable sequence of 20 nucleotides that will be complementary to the bacteriophage sequence. And then there is also the tracker RNA, which is a constant RNA sequence that forms a complex with the CRRNA. When the three parts are together, the CRRNA, tracker RNA, and the Cas9 protein, a cut will be induced at exactly 3 nucleotides from the panel. Of course, since this is a war, there are countermeasures that are taken by the phage. They will mutate their genome so that they become resistant. They are not recognized by the CRRNA and will survive bacterial defense. The CRISPR-Cas9 Big Bang started in 2012 when Jacques and colleagues realized that the CRISPR-Cas9 technology can permit to cut the genome of plants and animals. They also fuse the CRRNA and the tracker RNA together to form a single guide RNA. This was done essentially because the single guide RNA is something that can be patented, whereas the CRRNA and the tracker RNA, being natural, they cannot be patented. During the following year, there was an explosion of articles confirming that the CRISPR-Cas9 technology may be used to cut the genome of bacteria, human cells, farm animal, mouse, frog, zebrafish, plant, flies, nematode, yeast, monkeys. Many of these articles were published in prestigious journals such as Nature, Science, Nature Biotechnology, Molecular Cell, Cell, Molecular Cell. There was a rapid explosion of articles, and as you can see, as of November 26, 2020, 21,420 articles have been published where the word CRISPR is mentioned. The main reason of this rapid explosion of articles is that it costs much less to derive a sequence that will be able to cut a specific genome sequence compared to other pre-existing technology such as zinc finger protein and TALENs. As mentioned before, the binding of the Cas9 protein to the DNA require the presence of a photospacer adjacent unzip PAM, which is NJJ for the Cas9 derived from Streptococcus pyogenes. There is then the binding also of the single guide RNA, which will recognize a sequence of 20 nucleotides. And when the complex is made between the Cas9 protein, the single guide RNA, and the DNA, there is a cut that will be made at exactly 3 nucleotides from the PAM.
Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version.
One of the reasons of the rapid explosion is that the CRISPR-Cas9 technology also used all what was developed by gene therapy in the 30 years that preceded it. We have methods of delivering the DNA using AAV, adeno-associated virus, cationic polymers, liposome, microinjection, electroporation. All of those techniques are used in the context of CRISPR. When a double-strand break is produced in the DNA, most of the breaks will be repaired by non-homologous enjoining that will lead to micro-deletion or micro-insertion. And this is frequently used to knock out the expression of a gene by changing the reading frame. And it can also be repaired by homology-directed repair, which require the presence of a donor DNA that contains sequence of homology with what is preceding, what is following the cut, and in between the two homology sequence, there is a sequence in blue which may be a single nucleotide or a whole gene that will be inserted at the site of the double-strand break. When we use the CRISPR-Cas9 technology, we intend to produce a double-strand break at a precise site in the human genome. However, the Cas9 may sometimes induce cut at other sites in the genome, which are called off-target mutation. Computer software may predict these off-target mutations by using the whole human genome sequence. However, these softwares are not perfect, and they will sometimes predict off-target mutation at site where there is no cut, and sometimes there will be a cut at a site which is not predicted by the computer. There are, however, some experimental techniques, such as the guide-seq method, which permit to experimentally identify the sites of off-target mutation. This guide-seq technique used the introduction of a short 34 base pair oligonucleotide at the sites of the cuts, and later by PCR, the site of insertion may be identified by sequencing. The presence of these off-target mutations is the main problem that delays the use of CRISPR-Cas9 technology for direct in vivo corrections. One of the methods to reduce off-target mutation is simply to reduce by 2 or 3 the number of nucleotides of the single guide RNA which is binding with the target DNA. Another method is to use a mutated Cas9 nuclease that cut only one strand of DNA. It is then called a nickase. There is variation of Cas9 where the nickase cut only the lower strand of DNA, and other mutation of Cas9 which permit to cut only the upper strand of DNA. It is thus possible to induce more precise double-strand break in the DNA by using the Cas9 nickase and 2 single guide RNA, each detecting a sequence of 20 nucleotides close to one another. It is also possible to use a non-functional Cas9, a dead Cas9 nuclease, fused with the FOC1 nuclease. Then 2 guide RNA are again needed to detect sequences which are close to another, and the FOC1 nuclease need to form a dimer in order to be able to cut the DNA. Finally, some researchers have mutated the gene coding for Cas9. They have modified some sequence coding for amino acid to reduce the non-specific binding between the Cas9 and the DNA. The SpCas9 gene is too big to be inserted with a single guide RNA inside an adeno-associated virus, and thus researchers have identified Cas9 from other bacteria that are smaller. For example, the Staphylococcus aureus Cas9 is smaller than the SpCas9, but it has a different path which is more restrictive. Another type of CRISPR-Cas9 enzyme has been identified. It's called CPF1. It produces sticky ends. It's not a blunt cut of the DNA. There is thus a whole series of various Cas9 obtained from various bacteria that require different paths. The CRISPR-Cas9 technology may be used not only to induce cuts in the DNA, but it may also be used to induce the expression of a gene by fusing Cas9 with BP64, for example, or fusing Cas9 with CRAB to repress the expression of a gene. In my research group, we are using the CRISPR-Cas9 technology to develop therapies for various diseases, Friedreich ataxia, Duchenne muscular dystrophy, and Alzheimer's disease. I will present that now. We have initially used the CRISPR-Cas9 technology to develop a treatment for Friedreich ataxia. It is an hereditary disease due to the presence of a long trinucleotide repeat, GAA, in intron 1 of the frataxin gene. This long repeat reduced the expression of the frataxin gene, leading to neurological problems and cardiac problems. We have thus used the CRISPR-Cas9 technology and generated single guide RNA able to cut in intron 1 before and after the trinucleotide repeat, leading to its removal and increasing the expression of frataxin. Indeed, the removal of the trinucleotide repeat in cells of the YG8-SR mouse model of Friedreich ataxia doubled the expression of frataxin compared to the untreated cells and raised the expression of frataxin to almost the normal level. The SP-Cas9 gene is too big to be delivered with two single guide RNA by a single AAV. We are thus currently using the smaller CG-Cas9 gene to remove the trinucleotide repeat in intron 1 of the frataxin gene. Using this smaller CG-Cas9 nuclease, we are also able to remove the trinucleotide repeat in intron 1 of the frataxin gene. We are also using the CRISPR-Cas9 technology to develop a treatment for Duchenne muscular dystrophy. Duchenne muscular dystrophy is due to mutation in a gene coding for the dystrophin protein. It is a large gene containing 79 exons. Some of these exons do not contain a multiple of 3 nucleotides, and thus 70% of Duchenne muscular dystrophy cases are due to deletion of one or several exons, and the total number of coding nucleotides which are deleted is not a multiple of 3. For example, in this presentation, we have a deletion of exon 50 that does not contain a multiple of 3 nucleotides. This leads to a frame shift, and there will be a stop codon in exon 51. Thus, the beginning of the dystrophin protein is expressed, but not the end of the dystrophin protein. This situation may be corrected by inducing the skipping, that is the removal, of exon 51 in the messenger RNA. This is done using antisense oligonucleotide. This exon 51 may also be deleted by inducing cuts in intron 50 and in intron 51 to completely remove that exon. This results in the expression of the beginning of the dystrophin protein and of the end of the dystrophin protein, but there is, however, a small part of the protein which is missing in the center of the protein. The removal of one or several complete exons may thus restore the reading frame and convert a Duchenne patient into a Becker patient. However, some Becker patients have severe symptoms and are bound to a wheelchair at the age of 11. Therefore, the improvement of some Duchenne muscular dystrophy may not be significant. This is because the dystrophin protein has a complex structure. Indeed, in its central part, the dystrophin protein contains 24 spectrin-like repeats. Each spectrin-like repeat is made of 3 alpha helix, helix A, helix B, and helix C. Note that helix A is starting on the left side and helix C is ending on the right side. And normally there is a succession of ABC, ABC, ABC. The main problem of deleting complete exon to restore the expression of dystrophin protein is that the beginning and the end of the spectrin-like repeat, indicated in this scheme by the black arrow, do not correspond with the beginning and the end of exons. And thus, when removing complete exons, the resulting of the spectrin-like repeat structure may not be normal. Becker muscular dystrophy patients have a deletion of one or several exons, but the total number of coding nucleotides which are deleted is a multiple of 3 nucleotides, and thus there is no frame shift. This is the case, for example, for Becker patient having a deletion of exon 45 to 47 or having a deletion of 45 to 49. In both cases, there is no frame shift. The beginning and the end of the dystrophin protein are expressed. However, at the junction site between the remaining codons, there is an abnormal structure of the dystrophin protein, and due to this abnormal structure of the dystrophin protein, these Becker patients are bound to a wheelchair at an early age. Thus, in my research group, instead of trying to restore the normal reading frame by deleting complete exons, we have instead aimed at producing an hybrid exon, 5054, which not only restores the normal reading frame, but codes for a dystrophin protein with a normal structure. We have done our initial experiments using the myoblast of a Duchenne patient having a deletion of exon 51, 52, and 53. Thus, because the number of coding nucleotide was not a multiple of 3, there was a stop codon in exon 54 due to the frame shift. We have thus used the CRISPR-Cas9 technology to induce a cut in exon 50 and a cut in exon 54 to create the hybrid exon 5054. To produce this hybrid exon, we initially identify what are the possible SP-Cas9 PAM, or what are the sites where we can cut in exon 50 and in exon 54. We identified 10 PAM sites in exon 50 and 14 different PAM sites in exon 54. This table predicts what are the results of inducing cuts in exon 50 and in exon 54. When we have a blue square, it means that the cut has been produced in exon 50 right after the 3 nucleotide that codes for an amino acid. And the cut has been produced in exon 54 just before the 3 nucleotide that codes for an amino acid. Thus, at the junction point, we have amino acids that are, one is coded by exon 54, followed immediately by an amino acid coded by exon 54. There's no frame shift and there is no new amino acid produced. However, when the square is green, that means that we have at the junction site a new amino acid which is produced because we have cut in exon 50 right after one of the nucleotides of a codon. And the 2 nucleotides that will complete these codons are from exon 54. We have cut right after one nucleotide of the codon in exon 54, and thus there's 2 nucleotides left to create a new codon at the junction site. There's thus a new amino acid at the junction site, but there is no frame shift, and all the other amino acids are the correct ones. It is also possible with the square in red to have a new codon produced at the junction site, but this new codon is a stop codon. This is not what we want to do. All the squares in white are because there is a frame shift. We're cutting after one nucleotide of a codon in exon 50, and we're cutting before the last nucleotide of a codon in exon 54, and thus at the junction site, there is a new codon which is done because there is a frame shift, and all the amino acid that follows are not the correct one. We have thus tested various pairs of guide RNA. One guide RNA cutting in exon 50, and the other guide RNA cutting in exon 54. This produced an hybrid exon 5054 that had the predicted size of this hybrid exon. Note that when we are doing cuts in exon 50 and in exon 54 in a normal dystrophin gene, we are deleting 160,000 base pair. And despite that, the junction between exon 50 and 54 is of the size predicted. We have sequenced this hybrid exon that was produced by cutting in exon 50 and in exon 54. Not only were the hybrid exon exactly the size as predicted, but the sequence were also exactly as predicted. For example, we had at the junction site the fusions of exon 50 and exon 54 producing exactly the predicted amino acid. We also obtained at the junction site a new codon coding for the predicted amino acid or coding for a stop codon, and sometimes, as predicted by the white square, we had a frame shift, and when there was a frame shift, there were stop codons that were met in the resulting hybrid exons. As mentioned before, dystrophin protein contains in its central part 24 spectrin-like repeats, each one being made of 3 alpha helix A, B, and C. We have thus used a guide RNA to induce a cut in exon 50 in a sequence coding for helix C, and another guide RNA inducing a cut in exon 54, also in a sequence coding for helix C. The resulting hybrid exon 5054 codes for an hybrid helix C, where the beginning of helix C is coded by exon 50 and the end of helix C is coded by exon 54. This is a structure that has been computer predicted using the sequence of the resulting protein. When the myoblast of this Duchenne patient having a deletion of exon 51 to 53 are fused together to form some small muscle fibers in culture called myotubes. In fact, these muscle fibers do not express dystrophin because this is a Duchenne patient. However, the myoblast of a healthy human, when they fuse together, they form small myotube and they do express dystrophin, as can be seen here in this Western plot. When we use the myoblast of the Duchenne patient and we induce the formation of the hybrid exon 5054, we have expression of the dystrophin protein in culture. And as you can see, this dystrophin protein has a lower molecular weight than the normal dystrophin because there is deletion of exon 51 to 53 and an additional deletion of part of exon 50 and part of exon 54. Thus, there is a part of the dystrophin protein which is absent, and this is why the protein is smaller, but it is nevertheless expressed. For our initial in vivo experiment, we have used the HTMD mouse model. This is a transgenic mouse that expresses the human dystrophin gene, all introns and all exons. We have electroporated in the muscle of this mouse plasmid coding for the SpCas9 fused with the fluorescent green protein and 2 single guide RNAs. This, a month later, when we took the muscle, there was expression of the green fluorescent protein, confirming that the plasmid had been correctly electroporated in the muscle fibers, leading to the expression of the fluorescent green protein, and probably also leading to the expression of the SpCas9 and of the 2 guide RNA. We then extracted the DNA from these muscles, and we first confirmed using a test called the surveyor enzyme that indeed there were cuts that had been produced in exon 50 and 54. We then used PCR to amplify the hybrid exon 50-54. Note that in the muscles that are not treated with the CRISPR-Cas9 technology, there is no amplification of exon 50-54 because there are 160,000 base pairs between these 2 exons, and therefore the 2 primer for the PCR are too much separated from one another. We then sequenced this hybrid exon 50-54 that was produced in vivo in the mouse muscle expressing the human dystrophin gene. And as predicted, we had the sequence of exon 50 and a new codon at the junction site, followed by all the correct codon coding for the normal amino acid in exon 54. This is exactly as predicted in vivo. However, for delivery of the Cas9 gene in vivo to several muscles, we need to use an adeno-associated virus. As mentioned before, the SpCas9 gene is too big to be delivered with 2 guide RNA by a single AAV. And we therefore use for our next experiments the Cas9 of the Staphylococcus aureus, which is a smaller Cas9, which permit delivery with 2 guide RNA by a single AAV. In this case, we have used guide RNA that induce cuts in exon 47 in a sequence coding for helix B, and in exon 58 in a sequence again coding for helix B. This resulted in the formation of an hybrid exon 4758 coding for an hybrid helix B, which has a normal structure. The beginning of the helix B being coded by exon 47, and the end of that hybrid helix B being coded by exon 58. We then use for our in vivo experiment a new mouse model called HDMD delta 52. It's the same mouse model as previously that contains the dystrophin gene with all the exons and all the intron, except that this mouse has a deletion of exon 52, and thus there is no expression of the human dystrophin gene. We have thus used the CRISPR-Cas9 technology to induce a cut in exon 47 and a cut in exon 58, producing the hybrid exon 4758, resulting in the production of a dystrophin protein containing an hybrid exon, hybrid helix B. We have injected to these HDMD delta 52 mouse AAVs coding for SaCas9 and 2 single guide RNA. A month later, we observed in the muscle the expression of the human dystrophin gene, including in the heart. We are thus proposing a treatment of Duchenne muscular dystrophy, which would be the systemic delivery by an adeno-associated virus of the Cas9 gene and of 2 guide RNA to form an hybrid exon. In contrast with exon skipping, which is a treatment done at the level of the messenger RNA, the treatment that we propose at the level of the DNA would be permanent. The CRISPR-Cas9 technology is evolving rapidly, and new technologies derived from the CRISPR-Cas9 permits now the modification of a single nucleotide. More than 32,000 single nucleotide modifications are responsible for hereditary disease, and thus the capacity to correct a single nucleotide would provide treatment for most of these hereditary disease. The first treatment which permit the modification of a single nucleotide was developed by Kamar et al. In 2017. It used a Cas9 nickase, that is a modified Cas9, which is able to cut only one strand of DNA, and this Cas9 nickase is fused with a cytidine deaminase. This technology permits to chemically modify cytidine into a uridine, which is replaced by a thymine following DNA repair. The main limitation of that technique is the chemical modification of the cytidine will occur in a narrow window of 5 nucleotides located at 12 to 16 base pair from the NGG pan. We have initially technology to develop a treatment for Alzheimer's disease. The Alzheimer's disease is produced by an abnormal metabolism of the amyloid precursor protein. Normally, this protein is cut by the alpha secretase, followed by a cut by the gamma secretase. This produces peptides and protein fragments that are degraded without causing any problem. However, this protein may also be cut by the beta secretase, followed by a cut by the gamma secretase. And this produces short 42 amino acid long beta amyloid peptides that aggregates to one another, forming amyloid plaques that interfere with synaptic transmission, leading to neuron death and memory problems. This scheme illustrates the amino acid sequence of the transmembrane part of the amyloid precursor protein. We can see the position of the beta, alpha, and gamma secretase cut site. All the amino acids with the star above their name are amino acids which are modified, leading to familial form of Alzheimer's disease. Please note the position with the red arrow. This is the alanine in position 673. When this alanine is changed by valine, this leads to severe early onset Alzheimer's disease, and you are Alzheimer at the age of 40. However, when this alanine is changed by threonine, you're not Alzheimer, even when you're 95, 100, or 105 years old, as shown by Johnson et al. In Nature 2012. The presence of the A673T mutation, also known as the Icelandic mutation, reduces the secretion of A-beta-40 and A-beta-42 peptide for the wild-type APP gene and for APP gene containing the London mutation. Our experiments have shown that the presence of the A673T mutation reduced the secretion of A-beta-40 and A-beta-42 peptide by the APP genes, not only for the wild-type gene, but also for APP genes containing several familial Alzheimer's disease mutations. As mentioned before, the CRISPR-Cas9 base editing technology permits to modify the cytidine into a thymine. We thus have used the base editing technology to target the cytidine in the antisense strand of the alanine codon, transforming that cytidine into a thymine and just changing the alanine codon into a threonine. The main problem with this approach is that although we want to modify the cytidine into the antisense strand of the alanine codon, there are other cytidine nucleotides nearby that are also affected by the base editing approach. We have constructed 14 different base editing enzymes to be able to modify more specifically cytidine in the antisense codon of alanine. By modifying the cytidine in the antisense codon of alanine, we have been able to introduce the A673T mutation in up to 17% of the APP gene. However, other cytidine, also located in the antisense strand, were also modified by this approach. A new fantastic technology called Prime has recently been developed by Antelon et al. It permits, in principle, to replace any nucleotide by any other nucleotide. The prime editing technology uses a Cas9 nickase fused with a reverse transcriptase. It also requires a prime editing guide RNA known as a PEG RNA. The PEG RNA is essentially a prolonged single guide RNA. Thus, as the single guide RNA, it contains a spacer sequence which react with 20 nucleotides in the target DNA. It also contains the constant scaffold of the single guide RNA, which is in red, and then at its 5' end, there is a prolongation with the primer binding site, which is a sequence of 10 to 17 nucleotides reacting with the upper strand of DNA. This is followed by the reverse transcriptase template, again, which is 10 to 17 nucleotides in length, and which will contain some modified nucleotide, in red in this case, indicating which nucleotide has to be modified by the reverse transcriptase. A plasmid designed by Anzalone et al. Is available at Adgene to construct new PEG RNA. This plasmid contained the red fluorescent protein gene, which may be removed by BSA1 cuts. This produces a backbone of the protein, and then the other components are the spacer, the primer binding site, the reverse transcriptase template, and the PEG RNA scaffold. All of these sequences are single-stranded oligonucleotides that may be purchased from IDT. These 4 parts are then assembled together to produce a new PEG RNA. To use the prime editing technology, we first have to identify photospacer adjacent motif, PAM, which is NJJ for the Cas9 of Streptococcus pyogenes. This will permit to the Cas9 to bind to the DNA. Then the spacer sequence of the PEG RNA will bind to a 20 nucleotide sequence of DNA, in this case the lower strand, and the formation of the complex between the PEG RNA, the Cas9 nickase, and the DNA will induce a nick at exactly 3 nucleotides from the PAM in the upper strand of DNA. This will release the upper strand of DNA to be able to interact with the primer binding side of the PEG RNA. And then the reverse transcriptase template, which contains a few nucleotides to be mutated, will be available for the reverse transcriptase to synthesize a new DNA upper strand. Many Duchenne muscular dystrophy patients have a stop codon in the DMD gene. Since we did not have access to cells of patients containing such point mutation, we decided to introduce these stop codon using the prime editing technology. So in each case, we had to identify an NGG PAM, identify the PEG RNA photospacer sequence, and then modify the reverse transcriptase template so as to modify a codon for an amino acid into a stop codon. In this case, we have changed cytidine to thymine to introduce the TGA stop codon. We have successfully used that approach to introduce stop codon in exon 9, 20, 35, 43, 55, and 61. We have thus designed various PEG RNA targeting DMD exon 35. As you can see, we have varied the reverse transcriptase template in blue from 10 to 16 nucleotide, and we have also varied primer binding site in green from 10 to 15 nucleotide. And at the desired mutation site, we have introduced a T to introduce that mutation. Our initial experiments were done in HEK293 T cells. we tried to reproduce the mutation of MX1 gene and to target DMD exon 35. So essentially, these cells were transfected with plasmid coding for the Cas9 nickase fused with the reverse transcriptase and a PEG RNA targeting either exon 35 of the DMD gene or the EMX1 gene. The DNA was extracted 3 days later, and the targeted sequence was PCR amplified and sequenced using Sanger method. The sequence were analyzed using the EDIT-R online program. Essentially, we observed a 32% correction of mutation of the AMX1 gene as done by Enzalone et al. However, for the exon 35 mutation, we had a 2% background in the sequence of the control, negative control. And with the different PEG RNA, we have a mutation ranging from 4 to 8%. So this was not as great as for the EMX1 gene. We have thus tried different methods to try to increase the percentage of genome editing of exon 35. The first method that we have tried is to repeat the treatment 3 times. Essentially, the cells were transfected with plasmid at day 0, 6, 12. And DNA was extracted 3 days and 6 days after each treatment. We have amplified by PCR exon 35 at each of the extraction day and sequenced it by Sanger and analyzed the sequence. As you can see, the percentage of genome editing increased from day 3 to day 18 with the repeated treatment. And this was the case for all 3 PEG RNA that were targeting exon 35. We have then tested a second method to try to increase the percentage of mutation in exon 35. It is to induce a second nick in the target gene. This is the Pe3 method. Essentially, we have identified 2 PAM sequences which permitted to guide RNA to induce a second nick at either 57 nucleotide from the original PEG RNA nick or at 24 nucleotide from the PEG RNA nick. When we induced a second nick at 24 nucleotide or at 57 nucleotide from the nick induced by the PEG RNA, there was a significant increase in the addition of the target gene for PEG RNA 35.4 and PEG RNA 35.6, but not for PEG RNA 35.5. We still have to understand why. A third method to improve the percentage of mutation in the target gene is to mutate the PAM used by the PEG RNA. We thus designed PEG RNA that were not only able to introduce a stop codon mutation, but were also able to mutate simultaneously the PAM that they use. Mutation of the PAM used by the PEG RNA improved the percentage of mutation in the stop codon for 2 of the 3 PEG RNA that we have tested. Combining the 2 methods, that is, inducing a second nick in the target gene and mutating the PAM used by the PEG RNA, further increased the mutation of DMD exon 35 to 39% with all 3 PEG that we have tested. We are currently starting new project to correct mutation responsible for other hereditary disease. We are working on cystic fibrosis due to mutation in the CFTR chloride channel, on congenital muscular dystrophy due to mutation in the ryanodine receptor, and on spastic ataxia 8 due to mutation in the NKX6 type 2 gene. For each hereditary disease due to a point mutation, for example, here with spastic ataxia type 8, it is possible to correct, in principle, the mutated gene using the prime editing technology. In this case here, we can identify that the mutation is an adenosine change for thymine, and thus we can identify a PAM NJG for the SP-Cas9, which is close to the mutated nucleotide. We can then design a PEG RNA that will introduce the desired mutation. In this case, we are introducing two mutations. One, to correct the mutation, to reverse the thymidine into an adenosine. And the second point mutation is to modify the PAM so that following the correction, the Cas9 enzyme can no longer bind to the DNA. CRISPR-Cas9 derived technologies may thus be used to treat many hereditary diseases. The main problem remains the in vivo delivery of the editing agents. Thank you for your attention.
Okay, thank you, Professor Jacques. I'll take the turn now to make a general comment for those who don't speak English. Okay, thank you for your presentation.
Thank you.
Of course. Boa noite a todos. Meu nome é Matheus. Eu trabalhei como professor de inglês durante uns 2 anos e assisti a palestra junto com vocês e vou tentar passar um apanhado geral do que o professor falou. É claro que em poucos minutos eu não consigo passar a ideia e mesmo se eu tivesse todo o tempo que ele teve para poder fazer essa apresentação, eu não conseguiria passar porque eu não tenho a mesma expertise dele. Então, ele começou falando sobre a tecnologia CRISPR, Cas9, assim mesmo se pronuncia. Eu me adianto e peço desculpas pelos meus erros de pronúncia com relação aos termos biológicos, porque eu não sou da área, não conheço muito bem, mas vou tentar ajudar vocês a entenderem de uma forma geral. Podem comentar aí no chat, caso vocês não estejam entendendo, e me pedir que eu possa repetir alguma coisa. Então, a tecnologia do CRISPR, Cas9 é a derivada de pesquisas com bactérias, como ele mostrou nos slides, e somente em 2013 é que foi descoberto que essa tecnologia pode ser usada para induzir modificações físicas no genoma humano, e não só humano, como de outros animais, também foi mostrado por ele. E isso gerou uma grande explosão de artigos e pesquisas na área. Por que gerou essa grande explosão? Porque ao mesmo tempo em que um RNA guia, que foi o termo que ele usou, um único RNA guia para fazer um processo, é algo que pode ser patenteado quando se usa um processo natural, com múltiplos trabalhos de RNA, ele usou o termo RNA-CR e RNA-trans-CR, são processos naturais e que não podem ser patenteados. Então, passa a custar muito menos derivar uma sequência que será capaz de cortar uma parte específica do genoma, seja humano, seja de outros animais, em comparação com outras tecnologias pré-existentes, como a da proteínas NF ou do TALEN. Ele continuou dizendo que, quando eles usam essa tecnologia, eles pretendem produzir uma quebra da dupla parte, da dupla camada do DNA humano, em um lugar preciso. Eles especificam qual que é o lugar em que aquilo vai acontecer. Mas o que pode acontecer é que a Cas9 pode, às vezes, induzir um corte em outros lugares do genoma. E isso é o que ele chamou de limitação off-target, limitação fora de alvo, se eu não estiver enganado, fazendo uma tradução bem literal. Então, a presença dessas mutações ou limitações fora de alvo é o principal problema que atrasa aplicar essa tecnologia para as correções diretas in vitro. Continuando aqui, ele fala sobre o uso da tecnologia Crispr com Cas9, que pode não ser apenas para induzir cortes no DNA, mas também pode ser usada para induzir a expressão de um gene, usando o Cas9 com a VP64, por exemplo, ou fundindo o Cas9 com o CRAB, KRAB, para reprimir a expressão de um gene, dependendo do tipo de doença que está sendo analisada. Então, no grupo de pesquisa dele, eles estão estudando o uso dessa tecnologia para desenvolver terapias de várias doenças. Ele citou a ataxia de Friedreich, a distrofia muscular de Duchenne, a doença de Alzheimer, e ele apresentou um pouco sobre o uso nesses tipos de doenças. No caso da distrofia muscular de Duchenne, 70% dos casos dessa distrofia, eles são devidos a ter exons deletados, e o número total de nucleotídeos codificados que estão deletados geralmente não é um múltiplo de 3, o que causa um conflito na cadeia, como ele mostrou nas figuras. Assim, é possível corrigir esse salto fazendo a remoção do exon 51 usando o RNA mensageiro. Isso foi feito com oligonucleotídeos. Este exon 51 também pode ser eliminado induzindo cortes nas suas bordas para remover completamente ele. Então, a remoção de um ou vários exons podem restaurar o quadro de leitura e converter um paciente da distrofia muscular de Duchenne em um paciente de Becker. Portanto, a melhora de uma distrofia muscular de Duchenne pode não ser significativa, dado que um paciente com síndrome de Becker pode estar preso a uma cadeira de rodas, foi um dos exemplos que ele citou. No grupo de pesquisa, em vez de tentar restaurar o quadro de leitura normal e excluir esses exons complexos, no grupo de pesquisa dele, ele tem o objetivo de produzir um exon híbrido que faria a ligação das proteínas do exon 50 ao exon 54. E ele mostrou também uma tabela que consegue predizer quais são os resultados esperados. Eles fizeram todo o processo e fizeram a indução em ratos de laboratório, e a sequência do exon híbrido 50 e 54 foi produzida em vivo, ou seja, dentro do músculo dos ratos, foi exatamente igual ao que era previsto. Isso representou um resultado muito positivo para o tipo de pesquisa. Finalmente, ele também explicou o uso da pesquisa dele, dessa tecnologia, para o desenvolvimento de pesquisas em relação ao Alzheimer, e o principal problema relacionado à abordagem de corte, como ele vinha falando do uso dessa tecnologia que veio através das bactérias, o principal problema com essa abordagem é que, embora desejamos modificar a citosina na fita, existem outros nucleotídeos de citosina muito próximos que vão acabar sendo modificados também. E isso conseguiu ser vencido através da tecnologia de edição prime com a Cas9 nickase, que foi apresentada por um grupo de pesquisa, a Anzalone e os seus colaboradores que apresentaram isso. De uma forma geral, ele conclui dizendo que as tecnologias derivadas do CRISPR com o Cas9 podem ser usadas para tratar muitas doenças hereditárias. Inclusive, eles estão entrando em trabalhos com a fibrose cística e a doença muscular congênita, entre outras doenças. E o principal problema permanece na entrega em vivo. Qual que vai ser o desenvolvimento disso em seres vivos? Então, eu agradeço pela paciência de vocês para essa retomada geral dos comentários e espero que eu tenha ajudado em alguma coisa.
Obrigado. Professor Jacques, he summarizes your speech. Okay? Have the sound.
Yes, thank you very much for the translation of my summary in Portuguese of my talk.
Ok. Vocês terem uma ideia da importância dessa pesquisa, o laboratório do Dr. Jacques recebeu agora uma bolsa do governo canadense de $1,6 milhão para esse tipo de pesquisa do Crispr, utilizando na terapia de doenças. Nós estamos trabalhando com ele, também aqui no Brasil, e junto com a Fiocruz do Rio de Janeiro, e nós já estamos validando um novo teste para detecção de vírus, que poderá ser usado também para a Covid-19. Esse teste está sendo validado lá no laboratório junto com a gente, o professor, na Fiocruz do Rio de Janeiro, e ele detecta a presença do RNA viral em 2 horas. E é um teste feito em fita, não precisa de aparelhagem muito cara, pode ser feito em qualquer lugar dos países, mesmo no interior, e por qualquer pessoa, não precisa ser um técnico de laboratório, ele pode ser treinado, e nós recebemos aqui no Brasil, para essa técnica, 300 mil, e uma parte também veio da FAPERJ. E agora eu vou convidar o Dr. Gabriel Lamothe. O Dr. Gabriel Lamothe, ele é um mestrando do laboratório do Jacques Tremblay, e ele é o mestrando que obteve a maior bolsa de pesquisa de todo o Canadá. Dr. Gabriel Lamothe, it's your turn now. Welcome.
Thank you for having me. Let's see if we can get it started.
Yeah.
Can you see it? Hi, my name is Gabriel Lamothe, and today I'll be discussing the detection of SARS-CoV-2 using Crispr technology, namely the test that we've been developing in Dr. Tremblay's laboratory. Studies have confirmed that many individuals infected with SARS-CoV-2 virus are asymptomatic carriers.
Gabriel, you can't hear yet. No.
Yes, can you hear it?
No, the sound is very bad, then, Gabriel. Maybe you should talk.
Okay, I can actually try streaming it one other way, if that's the case. So, okay. Hi, my name is Gabriel Lamothe, and today I'll be discussing the detection of SARS-CoV.
Yeah, that's much better.
Is the sound better now? Using CRISPR technology. Oh, perfect. Namely, the test that we've been developing in Dr. Tremblay's laboratory. Studies have confirmed that many individuals infected with the SARS-CoV-2 virus are asymptomatic carriers of the virus. This is problematic in that the pandemic might be growing without adequate supervision. Simply focusing the testing on individuals with symptoms as opposed to entire populations is not enough. To better contain the virus until a large-scale distribution of an effective vaccine is underway, large-scale and continuous testing is mandatory. The current diagnostic pipeline for detecting SARS-CoV-2 is a 3-step process. The first step is to obtain nasopharyngeal swabs from patients. The next step is to isolate the total RNA from said samples and provide the RNA required for this third step, which is to use real-time quantitative PCR machines. The RNA is first reverse transcribed into DNA and then amplified. A dye is added to the amplification reaction to signal the presence of DNA strands. After a certain number of amplification cycles, a positive result will give a strong enough signal that the machine flags it as containing the original virus. Now, this process is problematic for a few reasons. The first is that real-time PCR machines are extremely expensive. Most can range from $15,000 US to well over $90,000 US. Given that it's a fairly specialized piece of equipment and that not all molecular biology laboratories need one, some get along just fine with a regular PCR machine, which costs a much more reasonable $5,000. As such, widespread testing for the coronavirus using these machines isn't ideal. Acquiring the large number of machines required for widespread testing is a feat in and of itself, and is likely one of the contributing factors for the sometimes long wait times for results. Many reports have stated that it can take 3 to 24 hours or more to obtain results in a clinical diagnostic laboratory. Over here in Canada, Ontario reports wait times of up to 4 days to get results. High-density metropolitan areas like New York are also subject to long lines for testing, and then a time to results of about 7 days for some clinics in that city. The creation of new types of detection tests is therefore an important step in curbing the spread of this virus. An ideal test needs to follow the World Health Organization's criteria for assured. That is, the test needs to be affordable for as many people at risk of infection as possible. Tests that require extremely expensive reagents or machinery are not ideal. The test must also be specific to avoid false negatives. That way, anyone who could potentially spread the virus can be identified and put into quarantine. The test must be sensitive so that few people who don't have the virus are mislabeled as having it and treated improperly. The test must also be user-friendly. That is, it must be simple to perform. And it must be rapid and robust. That is, it must give a time to results that is quick enough to make the most use from the results. Situations where it can take up to 7 days to obtain results are clearly far from ideal, as by the time the patient receives the information, they might have infected many others. The test must be equipment-free as much as possible. Tests that require too many rare and expensive machines aren't ideal for many countries. Finally, the test must be deliverable to those who need it. The discovery of Cas13, or C2C2, was revolutionary in the field of molecular detection. This class II, type 6 Crispr effector is similar to the protein Cas9, off of which base editing and prime editing were designed. This protein is also a member of the Crispr family. Unlike Cas9, however, Cas13 can be programmed to target RNAs as opposed to DNAs. By binding to its Crispr RNA, Cas13 is primed to target extremely specific RNA sequences. After locating a target sequence, the Cas13 protein is activated. This causes the protein to begin to cleave everything in its surroundings in an event called collateral cleavage. It cleaves its original target and then moves on to start cleaving all surrounding RNAs. Researchers realized that this collateral cleavage could be used to turn the protein into a switch for a detection test. Once the target molecule is present, the switch is flipped, the Cas13 activated, and all surrounding RNAs are degraded. By tagging certain RNAs in the solution with a fluorescent molecule and a quencher, it would therefore become possible to resolve a signal. Researchers therefore started to develop a detection test based on this protein for viruses, bacteria, and human diseases such as cancer. However, they soon noted that there were certain limitations to the protein and that its limit of detection was far from sensitive enough for their needs. Recently, certain tests have been designed using only Cas13. However, these tests are less sensitive and tend to use a different Cas13 protein. Researchers at the Broad Institute decided to supplement the protein with a few others that would amplify the target RNA. To this end, Dr. Zhang and his team created a new test called Specific High Sensitivity Enzymatic Reporter Unlocking, or SHERLOC for short. This test is based on the reverse transcription of target RNA into DNA. Afterwards, the DNA is amplified at a constant temperature by using a process called recombinase polymerase amplification. This technique amplifies a small amount of DNA at a constant temperature, unlike the more commonly used PCR. The incorporation of this step also means that the ensuing detection test can target DNA. Since the RPA reaction simply amplifies a determined section of DNA using two primers, the initial genetic sequence can either be RNA or DNA, so long as reverse transcriptase enzymes are present for RNA. The amplified sequences are then transcribed using a T7 RNA polymerase to create vast quantities of the target RNA. This then allows the Cas13 to have a far larger pool of target molecules, which can activate the enzyme. Once the enzyme is active, it does what we discussed before and starts to cleave all surrounding RNAs, including reporter RNAs, which give off the easily detectable signal, which makes the test work. In their initial version of the Sherlock test, two different reporter molecules were used. The first used a fluorescent molecule as well as a quencher, and it was able to produce a lot of fluorescence after cleavage of the RNA. The other was designed to interact with a few antibodies, and when it was cleaved, it appeared as a distinct second band on a strip that was quite similar to a pregnancy test. While this test was good in many ways, it had certain drawbacks. At the beginning of the pandemic, we attempted to replicate it and immediately had a few issues. The biggest issue by far was that some of the materials required for the test to work were only available in extremely limited supply. The kits required to perform RPA reactions, for example, took approximately 2 months to arrive, and even then, we only received enough materials to perform 200 tests. This clearly wasn't going to be enough to produce sufficient tests in the future. Additionally, the reporter that used a pregnancy-style strip was remarkable and seemed to be very useful. However, since it was too difficult to acquire in sufficient quantities, we decided not to use it and to instead focus our efforts on using the fluorescent-style reporter RNA. Since the original version of the test could not be used by us in the way that it was intended, we decided to redesign it. In redesigning the test, we had several options and routes that we could take. We knew that we wanted to keep on using Cas13 because we had a large supply of this enzyme due to an ongoing collaboration with Dr. Alain Garnier's lab. We also knew that we wanted at all costs to maintain the isothermal nature of this test. One of the biggest advantages to Sherlock was that it didn't require large amounts of expensive equipment. Simply by using a heating block or something of the sort, it was possible to get a visual readout. We therefore decided to employ loop-mediated isothermal amplification, or LAMP for short. This amplification strategy was fairly well documented and easy enough to perform. Based on the use of 6 primers, this technique could rapidly amplify large amounts of target DNA. Unlike both RPA and PCR, however, LAMP doesn't create identical amplicons of a discrete size. Instead, the 6 primers can interact to create far larger molecules of a repeating sequence. Initially, it was a little difficult to incorporate this isothermal amplification strategy into our test. As you might remember, the step following RPA in the original Sherlock test was one that produced lots and lots of target RNA. This production of target RNA was only possible because the RPA step added a tag that let the T7 RNA polymerase bind to the DNA and begin transcribing it. However, no one had seemingly ever done that with LAMP. And since this new technique used 6 primers, 2 of which looped back on themselves to create weird dumbbell-like structures, it was difficult to figure out exactly where to incorporate this tag. After a while, we decided to include the tag right in the middle of one of our looping primers. This has consistently given us very strong results. We were therefore able to retrofit the original Sherlock test with a new amplification strategy, all the while still benefiting from Cas13's specificity and signaling. The new test is therefore using RT-LAMP as opposed to RT-RPA. Of course, should we become interested in targeting DNA in the future for either certain types of viruses or certain bacteria, this test would be readily applicable to that as well. We also decided to use a fluorescent reporter RNA instead of the pregnancy-style one that we spoke about previously, because it seemed to be the most readily available material. Since it doesn't take any particularly advanced machinery to make it work, we were happy to choose this one. Now that you know how the test works from a theoretical standpoint, let's discuss it from a more practical point of view. For now, the test requires that we start with RNA that was extracted from a virus using a commercially available kit. 1 microliter of this solution is transferred over to the tube containing the RT-LAMP reaction. Ideally, in the future, we would like to modify the test so that it becomes possible to directly add a patient's saliva to it and continue on from there. That would noticeably cut down on the labor required to obtain each reading, cut down on the overall costs, and make it much faster for the results to come in. After transferring 1 microliter of the RNA extract to the first tube, the tube must then be incubated at 65 degrees Celsius for 30 minutes. After that, 1 microliter is transferred from the first tube into the second, which contains the T7 and Cas13 solution. After tapping the tube a few times to mix it and microcentrifuge it down for a few seconds, all you need to do is incubate that tube at 37 degrees Celsius for 30 minutes. At that point, all of the reactions are done and the results are ready to be read. By exposing the tubes to a wavelength of approximately 490 nanometers, it's possible to easily detect which samples contain SARS-CoV-2. Those ones will fluoresce a bright green. Those that didn't contain any of the viral specimens of interest will be completely colorless and indistinguishable from a tube filled with a little water. I'll show you that later. Now, it's worth noting that the DNA and RNA sequences that we've used in this first version of our test were all previously published by different groups. As such, we do not believe that they will result in a positive signal when in the presence of a relevant RNA. This is particularly important because your saliva is full of RNA. The extraction process for the virus does indeed result in viral RNAs being captured, but there tends to be a whole lot more human and bacterial RNAs too. In addition, other coronaviruses that are much less dangerous are fairly common in the human population. If our test gave positive signals every time someone came in with a common cold, then it wouldn't be very effective. As such, we have based ourselves on previously described templates to try and avoid this as much as possible. In designing this test, we tried to make sure that it wasn't overly reliant on complicated machines. We wanted the entire test to be easy to perform using only a few common laboratory apparatuses. As a result, with this current iteration of the test, all we need is one machine that can heat PCR-style tubes to 65 degrees Celsius, and one that can heat the PCR tubes to 37 degrees Celsius. We also need two either P2 or P10 pipettes for reasons we'll get into later, along with their associated pipette tips. A small microcentrifuge capable of taking PCR tubes is also required just to make sure that the reaction mixtures don't touch the top of the tube when you're opening them and potentially contaminating all of the surrounding tubes. Finally, a machine capable of giving off ultraviolet wavelengths is necessary. In this picture, you'll see the machine I've been using. It's most commonly used to visualize gels in a molecular biology lab, but it does a really good job of visualizing our positive samples in our test. For the two incubation periods, several different options are available. I've been using an old thermocycler to date, as I find it does a really good job of evenly heating my tubes. It's worth noting here that this means that even old and run-down thermocyclers that no longer do a very good job of cycling between temperatures can be readily used in this test. Unlike the current RT-qPCR gold standard that requires specialized real-time thermocyclers, here any old machine can be used. It's also possible to use other common laboratory apparatuses, such as block heaters, that have an adapter for PCR tubes. Please note that in this picture, the block heater has an adapter for 1.5 milliliter microcentrifuge tubes, so the true adapter will look a little different. I've also used incubators to great success. Even bacterial incubators that operate at a steady 37 degrees Celsius can perfectly suit the needs of this test. So long as the tubes are readily exposed to the ambient temperature, then positive signals should have no difficulty in resolving. Please note, however, that when using incubators, it's better to have the tubes exposed to the air. That way, they don't waste time as their rack slowly begins to heat up. Water baths are one final option that you can choose. Personally, I tend to avoid this option because I find the risk for cross-contamination between samples to be much greater and therefore unideal. However, I have tried to perform RT-LAMP reactions with this system, and so long as you're careful, it's very possible to do it. Once the reactions have been completed, it's possible to use various different machines to visualize the tubes depending on the materials your laboratory has. Personally, my machine of choice is a gel UV emitter. These machines, which are more commonly used to visualize agarose gels, do a very good job of safely showing which tubes are fluorescent and which aren't. Black lights can also be used for this test, however, I have found them to be slightly less desirable. Since the reporter RNA best absorbs wavelengths of 490 nanometers, I have found that black lights which produce wavelengths of 390 nanometers to be less favorable for this test. For them to work, it's better to visualize the tubes in a dark room. In both cases, the machines should really be placed in a darker room away from excessive light. A little background light seems to have less of an effect in the case of the gel UV light, however. This picture, seen on the left, was taken using a generic iPhone when the tubes were placed on the gel UV light. The lights in the room were only partially turned off, resulting in lots of ambient lighting, even if there were no lights directly pointed at the machine. Given the strong and even radiation that the tubes received on this machine, it's easy to determine which samples were positive and which were negative. In the case of the black light on the right, it's clear which samples are positive and which are negative, of course. However, the fluorescence is not even, and it's a little bit harder to detect by the naked eye. In this case, the camera on the iPhone took a very good picture and made it easier to visualize the tubes overall. Now, there are several important considerations to be had when using this test. For it to work properly and not give either false positives or false negatives, it's important to first designate proper working stations for each section of the test. It's also necessary to follow the predetermined reaction times without varying, otherwise the results might start to differ slightly. Finally, those handling the test must be careful not to contaminate any of the tubes with RNases. The first consideration to separate working areas is extremely important because lamb is excessively sensitive. This technique is known to produce lots and lots of concentrated target DNA. We, as well as others, have noted that when we open tubes that completed the lamp amplification in the same area that we originally used to set up the reaction, it's possible to contaminate the working area with aerosols containing the product. If you continue to set up the lamp reactions in those areas afterwards, then you run the risk of contaminating your new tubes with amplicons from the previous reactions. By touching your desk or various other contaminated surfaces with your gloves, you run the risk of artificially seeding your reaction mixtures that contain patient RNA with no SARS-CoV-2, and therefore producing positive results when there really shouldn't have been any. By separating the areas in which you set up the lamp reactions and those in which you use the amplified sequences, you run less of a risk for this to happen. It's also necessary to have designated lab coats, pipettes, and other such materials for each working station. If those in workstation 2 become contaminated, you don't want that coming back into workstation 1. Please note that this is much less stringent when bringing materials from workstation 1 to workstation 2. Since it's the RT lamp reaction that runs the greatest risk of contamination, bring material, for example, gloves, from that station to the next should not be a problem, provided that the gloves are still relatively clean. Please note that this situation of contamination via aerosol is of much less importance during the second stage of the test. As we discussed previously, the Cas13 protein we are using requires a relatively large amount of RNA to be functional. At the present time, we are not concerned in the least that an aerosol contamination from lamp might produce false positives of our Cas13 reactions, as we have yet to see a single situation where this has happened. The second consideration of note is that the reaction time must be followed. A small discrepancy can be tolerated, that is, 1 minute more or less doesn't really seem to have a large impact on the test. However, more than that can start to become problematic. In the case of the RT lamp reaction, external teams, as well as us, have seen that by doubling the reaction time for the lamp, false positives can arise. As you can see here in both pictures, a successful lamp reaction can also be visualized on an agarose gel. The successful reactions are characterized by the presence of several large bands of varying sizes. However, when the lamp reactions go on for too long, it's possible to begin amplifying samples with nothing in them. For example, on the left, you'll see a gel that was taken from an article in which at 30 minutes, the wells containing reactions with 10 copies of the target viral RNA and no copies of the target viral RNA had no bands. In the case of the gel on the right, a gel that I made, 3 of the wells contained reactions that had no target RNA to speak of. Only the N fragment well contained target RNA. At 30 minutes, these negative controls did not demonstrate any amplification, as we expected. However, at 60 minutes, both the published gel and mine started to give amplified products. In their case, The 10 and 0 copies both started to be amplified, and in my case, one of my negative samples started to get amplified. I've performed several experiments so far with 30-minute incubation times, and so far have never gotten false positives. That's not to say that it's entirely impossible, simply that it's very, very unlikely. As a side note, this is a good time to mention that changing gloves frequently when performing the first reaction is a good way to avoid these kind of unlikely false positives. After prolonged use, gloves might become contaminated with small amounts of viral RNA, and that could potentially lead to false positives even if we're only incubating at 30 minutes. The final consideration to look out for is the presence of RNases. For those of you who might not be familiar, RNases are enzymes that cleave all RNAs they come across. Both steps for this test are sensitive to RNases. The initial RT lamp begins its amplification off of RNA, so the presence of RNases could be very detrimental and therefore lead to false negatives. The second step of the test, the T7 and Cas13 step, uses an RNA reporter to signal positive tubes. If, while you're using the test, you start to introduce RNases, then you run the risk of getting false positives. RNases are extremely difficult to remove once they're introduced into a system, so the best means of protecting your test is by using prevention. Since your hands produce lots of RNases, it's mandatory to use gloves at all times when working with the test. In doing so, you will prevent your body's natural RNases from degrading the initial SARS-CoV-2 RNA and protect the fluorescent reporter at the end. Gloves must also be changed whenever they come into contact with skin, hair, or other frequently manipulated objects, such as doorknobs and personal devices. The use of RNase and DNase-free filtered pipettes is also important for this test. By using these kinds of specialized tips, you can make sure that any possible contaminations you might have in your pipettes doesn't have a chance to contaminate your test. In areas where the risk for RNase contamination is fairly high, it might be worthwhile to treat all surfaces or tools with an RNase removal solution. These kinds of solutions can degrade RNases that might otherwise stick to your gloves and contaminate your samples. Please note, I haven't had to use this solution very often. So long as you maintain a clean working schedule, you should be fine. The second component for the test, the one containing T7 and Cas13, is also designed to be able to tolerate a certain level of contaminating RNases. There is an RNase inhibitor added to the reaction mixture to prevent the degradation of the reporter in case the original SARS-CoV-2 RNA wasn't completely clean, or in case contaminations occurred while you were preparing the test. That said, the presence of excessive RNases can still fully activate the test. Seen here on the right, 1 microliter of very concentrated RNase A is capable of causing the test tube to become fluorescent. It's therefore important to take the required
Gabriel, on t'entend peu.
test that can be widely distributed while also meeting this limit of detection is fully functional for curbing the spread of this pandemic. While RT-qPCR is much more sensitive than this, its improved limit of detection might not necessarily be that beneficial. Since this technique can pick up RNAs freed by infected and dead cells after the infection has run its course, this form of screening can cause individuals who are nominally SARS-CoV-2 RNA positive, but aren't actually infectious, to be isolated unnecessarily. As you can see here on our left, our detection test is able to detect samples that have 100 copies per microliter, as was stipulated by the model. A more recent comparison between the different primers that we've been using for LAMP has demonstrated that one version of our test can detect samples containing only 80 copies per microliter, while the other can only detect samples containing 1,000. Moving forward, we're likely to focus our efforts on the first primer set to ensure that we meet the limit of detection that was stipulated by the model. As a final note, we're happy to report that our test works not only on fragments of the SARS-CoV-2 genome, it also works when exposed to the virus's full RNA profile. Initially, we began the development of our test by using fragments of the virus's genes that we had transcribed ourselves in the lab. Recently, we've used the test on actual SARS-CoV-2 viruses and have obtained the expected results. As you can see on the left, the first section of the reaction, the RT-LAMP reaction, works just as expected on the viral RNA. The top left wells that were run on the agarose gel clearly demonstrate a strong amplification. Below them, 3 reactions were performed with cells that had not been cultured with the virus. The viral RNAs were created by first isolating the viruses from a patient. These viruses were then cultured in Vero E6 cells, and after a few days, the culture medium was treated to extract any and all RNAs that were present. The viruses were therefore destroyed and their RNA was obtained. When the LAMP amplicons that had amplified the real virus were transferred over to the second stage of the test, they gave the expected strong fluorescent signals. At the moment, what remains to be done is to use our test on samples that have been confirmed positive or negative by RT-qPCR, the current gold standard in SARS-CoV-2 detection, and see how our test compares. This will give us a better indication of what our test's sensitivity and specificity are. Just to wrap up, I would like to thank Dr. Gary Kobinger, Dr. Alain Garnier, and Dr. Guy Boivin for having helped to provide finances or materials for this project. I'd also like to thank Dr. Tremblay and everyone in his team for having helped to provide certain insights as I was redesigning this test. Finally, I'd like to thank Dr. Marcello for having helped to organize this entire meeting, and the Canadian Institute for Health Research and the FRQS for having helped finance me as I pursued this project. Okay, so that was my presentation. Yeah.
Thank you, Gabriel. I'll take the time just to make to summarize it the best I can here for the non-English speakers. Thank you very much for your presentation.
Thank you.
Ok, vou fazer um resumo assim como eu tentei fazer com a apresentação do Dr. Jacques. Deixo bem claro que é um resumo de uma apresentação que carrega tanto conteúdo e tanta experiência de trabalho, não pode ser considerado assim uma experiência, como assistir a apresentação. Então, se vocês tiverem a oportunidade de voltar ao YouTube, pesquisar melhor os temas, de repente assistirem uma velocidade mais lenta, talvez possa ser bom para todos vocês terem uma experiência melhor com isso. De toda forma, eu vou tentar fazer um resumo do que o Gabriel falou. Ele mostrou o desenvolvimento da COVID, que todos nós estamos acompanhando. De uma forma geral, o teste atual se faz através de uma máquina de PCR em tempo real, e esse é um processo muito caro que demanda o processo em si não é caro, mas a obtenção das máquinas é muito caro e elas não são bem distribuídas no globo, ou seja, não são bem distribuídas em todos os lugares que necessitariam fazer esse teste. Assim, surge a necessidade de se criar um novo estilo de teste para que possa ser, de uma forma mais geral, aplicado em todo o globo e acessível a todas as pessoas, como recomendado pela Organização Mundial da Saúde. A descoberta da Cas13, ou C2C2, que é o que ele ressaltou, torna importante porque ela faz torna esse processo mais fácil e mais importante, uma vez que ela faz o mesmo processo que a Cas9 consegue fazer com DNA, só que ela faz com RNA. E dado essa característica de fazer com RNA, é possível isolar o RNA dos vírus para desenvolver um tipo de teste. E foi isso que eles fizeram. Alguns testes usando a Cas13 foram desenvolvidos, mas não foram muito seletivos, ou seja, não responderam muito bem a apenas aquele tipo de vírus, ou geraram algum tipo de falso positivo. Eles tentaram, então, desenvolver o estilo de teste que eles chamaram de Sherlock, que é um teste específico de alta sensibilidade enzimática, e esse tipo foi desenvolvido com 2 tipos de moléculas usadas durante esse processo. tentaram manter esse design do Sherlock, atualizando ele com uma amplificação isotérmica, que é o que ele chamou de LAMP, e assim fizeram o teste para a COVID usando o Cas13a e esse Sherlock LAMP, essas tecnologias. Basicamente, o teste consiste em tirar algo que teve contato com o vírus, alguma amostra que teve contato com o vírus, passar pelo processo de amplificação isotérmica e incubar ela por 30 minutos a 65 graus Celsius. Daí tira-se 1 microlitro dessa amostra e coloca esse microlitro em uma solução que contém a Cas13 e mantém a solução por 30 minutos em 37 graus Celsius. O resultado será mostrado através do uso da luz fluorescente, que vai mostrar a cor verde em caso de positivo. Tivemos alguns desenhos dos slides dele, onde é possível ver claramente esse procedimento do teste. É importante analisar o final do teste em um lugar onde não haja interferência de outros tipos de outras fontes de luz, porque poderia causar algum tipo de interferência na leitura do resultado final. De modo geral, o objetivo do teste é evitar que ele responda a outros tipos de coronavírus, já que existem outros tipos de coronavírus que não causam nenhum tipo de infecção tão letal ou tão ruim quanto o atual tipo de coronavírus que nós estamos enfrentando essa pandemia. O teste deve ser mantido o mais simples possível e, por isso, eles tentaram minimizar o uso de aparelhos. Então, ele mostrou na sua apresentação quão simples, relacionado ao estilo de teste atual, o teste que eles propõem pode ser. Então, a grande maioria dos aparelhos que ele mostrou são aparelhos comuns de laboratórios e não seria necessário uma máquina de PCR como é o estilo atual, dado que essa máquina é cara e tem pouca distribuição. Então, os aparelhos usados poderiam ser incubadores, aquecedores de blocos. Outras sugestões que ele faz para o uso desse teste é separar em diferentes áreas de trabalho cada teste ou cada conjunto de teste. Seria importante também seguir de forma estrita o tempo das reações, pois erros maiores que 1 minuto podem levar a falsos positivos, como eles observaram. E, finalmente, evitar contaminações de outros RNA vindos de fontes externas durante o processo de testagem. De uma forma geral, o teste pode ser usado com exemplos de RNA de vírus, numa forma geral, não precisa ser só para Covid-19, como é o caso da situação atual. Eu acho que eu fiz um apanhado bem geral dos dois e passo a palavra agora para o professor Jacques.
Thank you, Matheus, and thank you, Dr. Gabriel. Professor Jacques, would you like to talk something?
At this point, I have nothing to add, except if there is some question that I can answer. Otherwise, I thank you for organizing this session.
Thank you. Question, please? Please write in English. Can you read the comments,
Sorry?
Professor? Can you read the comments?
Yeah, but I think that
chat?
most of them are in Portuguese.
No, I asked you to write in English. I have asked.
In the comments. Oh, okay. I was looking at private chat. I'm going up the list. Oh boy, lots of comment.
Are you in commenting chat?
Yeah, I'm just looking at the comments very quickly.
Scroll up.
Anacrestia, is the probability that the CRISPR test will give false negative results? That's a question mostly for Gabriel. Gabriel, can you answer that?
So at the moment, we don't entirely know with my test. Generally, tests similar to this one have a sensitivity of about 90%, So the probability of giving a negative result is very low because it also tends to have a sensibility of about 80 to 90% as well. So overall, it's better than some of the alternatives that exist that aren't RT-qPCR.
Okay. Another question for you, Gabriel. It's also from Anna Cristina. Does the CRISPR test apply to other disease like Ebola, Zika, or dengue?
So we actually intend to adapt the test to Ebola as soon as we can. We're toying with the idea of doing Zika, LEPS, and a few others, because all we need to do is change a few of the sequences that we're using. So the LAMP primers and the guide for the CRISPR-Cas13 protein. And at that point, we'll immediately be able to start detecting those viruses as well. So now that the test exists, it's just a matter of optimizing it for every individual virus.
Okay, there's another question from France Antarctique Nouvelle. What are the perspectives of using the CRISPR-Cas9 technology to treat HIV? I must admit I'm not an expert of HIV. I know that some groups are working on that. I don't know what are the chances that whether or not this will work. But I think this is reasonable. You know, the bacteria have been using the CRISPR-Cas9 technology since millions of years to cut the virus. And so the idea here is to use the Cas9 technology to cut the HIV virus. I think this is quite reasonable. In fact, at the beginning of the COVID-19 pandemic, I saw a group in the United States that were proposing to use the CRISPR-Cas9 technology indeed to cut the COVID-19 virus. So it's the same approach for the HIV. I think this is a reasonable approach. My lab is not working on that approach. Also from France Antarctique, can this technology substitute the antibiotic in the near future, for example, to treat multi-resistant tuberculosis? Again, I think it's a good suggestion, and yes, indeed. The main problem with these CRISPR-Cas9 technology is to find an adequate technique to deliver either the Cas9 protein or the Cas9 gene. And so far in gene therapy, what is being used is the adeno-associated virus. Unfortunately, the adeno-associated virus costs a fortune to produce in good manufacturing practice. So we have to find another delivery method to deliver either the Cas9 protein or the Cas9 gene, and then it would ease using it, because right now it would be just too expensive to be used instead of an antibiotic. Also from France Antarctique, CRISPR-Cas9 may be an option in the near future to treat COVID, Ebola, Zika, malaria. Well, all of that I think is feasible. If I would have more money in my lab, we could start working on that also. I think these are reasonable projects. Many of the comments are in Portuguese, unfortunately.
There is no question in Portuguese. Only comments. There is no question.
Let me try to help. Is there any question in Portuguese?
There isn't.
There's
only comments.
Yeah. I see that I vary between 2 and 5 stars. One question from Vitor. I'd like to know if CRISPR is affordable to public health system, and if it can contribute to the cost reducing of genetic therapy that already exists. The main cost of gene therapy that currently exists is the production of the adeno-associated virus in good manufacturing practice condition. The last time I looked into doing a clinical trial for systemic delivery of some gene therapy, it was about $1 million to produce enough AAV to treat one patient. So to do a clinical trial on 10 patients, you needed just to start a budget of $10 million. And of course, so far, I have not been able to obtain such large budget. The only people that have such large budget are American companies that will spend a fortune doing these clinical trials, and they have millions of dollars available. The problem is indeed that after the treatment has been demonstrated effective, the cost of the treatment is $2 to $3 million per person. So I think it's the future. We need to develop gene therapy for many hereditary disease, but we also need to develop a cheap gene therapy. I think ideally gene therapy should be around $100,000, not $1 million, because most of the public health system cannot afford to do that. I think that it's only in United States where rich people can afford to pay for those, and poor people will not get access to the treatment. Still looking down for the list to see if there is any other question.
Excuse me, Professor Jacques. Just to make a small comment here, for those who doesn't speak English, I'll try to get the general idea from the answers you just gave, and Gabriel also. Just a second. As perguntas principais aqui foram se esse estilo de tratamento, ou esse estilo de teste poderia ser usado para outras doenças, como citaram Ebola, citaram HIV. Eu não estou muito bem lembrado das outras. Vocês se lembram, Bárbara, Werber-Bandeira? Tuberculose, sim. Zika vírus.
A substituição dos antibióticos pela técnica Crispr.
Sim. Então, de uma forma geral, o teste pode sim ser abrangido para outras doenças, mas não necessariamente vai ser realizado agora, e precisaria de um certo investimento e um certo tempo para ser feito. Do ponto de vista do uso da tecnologia para curar algumas doenças, ele acha que é bem possível que seja desenvolvido, o professor Jacques disse isso, mas que precisaria de um investimento muito maior para que isso acontecesse, de outras pessoas também pesquisando e com o tempo. A última pergunta foi uma das mais interessantes, se seria o caso de esse tipo de tratamento ajudar, se ele custaria o suficiente para que um sistema público de saúde pudesse pagar para uma população grande, e o professor Jacques esclareceu que se houvessem laboratórios públicos desenvolvendo a tecnologia, sim, mas que provavelmente essas tecnologias vão ser desenvolvidas por grandes farmacêuticas que vão cobrar muito caro por esse processo, que talvez no futuro, não para agora, mas no futuro, seja acessível sim ao público. Okay, Dr. Jacques, thank you for your time, and is there any question, you can keep reading and answer them.
Well, eventually, if there are other questions, people can always send me some emails.
Okay. Thank you, Professor Jacques Tremblay, for this wonderful meeting. Thank you, Gabriel. Thank you, Dr. Barbara Oliveira. Thank you, Matheus Chaves. And I hope you have another one meeting like this. Thank you very much.
Thank you.
Good night.
Thank you.
Thank you.
Have a good night.
Bye. Thank you.
Gostaria de agradecer a todos, em nome da FAF, pelas apresentações. Vou deixar para vocês um link lá no chat, para quem quiser emitir certificado. Esse link está disponível até às 23 horas, e é só acessá-lo e recebe o certificado por e-mail. Agradeço novamente, professor Bandeira, Matheus, professor Jacques e Gabriel, que já saíram da sala, mas agradeço a presença de todos e todos os que colaboraram com esse evento. Espero que seja o primeiro de muitos. Um grande abraço a todos e boa noite.
Boa noite.
Até mais, boa noite, um abraço.
Boa noite. Obrigado, Bárbara.
Obrigado pela oportunidade.